here's what happens on the end of the chromosome. Here's your chromosome, right? And the end of the chromosome is not, the DNA looks like this. There's not enough room to bind and replicate. There's the repeat we talked about. The repeat we saw was TTA-GGG, TTA-GGG, over and over again. And that's on the DNA. This is the DNA strand here, right? This guy here is the DNA strand on the end of that DNA there. This blue with this thing here is the ribonucleoprotein particle. This is the enzyme telomerase. The RNA that's in there has some of these repeats that are complementary. So this TTA-GGG would be AAU-CCC. The telomerase lines up with the DNA because of the complementarity, so as it does there. And now DNA telomerase can go and it can copy this. Remember, this is the 3' end of the DNA, so it can use this as a template and it can extend this piece of DNA. There's the red being extended. So it uses the RNA in the telomerase molecule, and the telomerase is the enzyme that actually puts these together. What kind of a polymerase is, what do we call this enzyme telomerase? How would we define it as opposed to a DNA polymerase? I guess I don't know how to ask this question. So DNA polymerases, what are they used for a template? DNA. So they're DNA-dependent DNA polymerases, right? This guy, telomerase, what's it using as a template? That RNA is in there, right? So these are RNA-dependent DNA polymerases. Right? RNA-dependent DNA polymerases. What does that look like? What's that? What other enzymes like that?
Reverse transcriptase.
Reverse transcriptase in viruses, and I'll show you that in a minute. Okay, so this is like reverse transcriptase, and it looks very similar. What happens now is once this strand is extended, right, and this guy lets loose, then the elongated DNA can serve as a template for synthesizing more DNA on the bottom. Right? So you extend the 3' strand this way, and then it's long enough that it can be primed and synthesized back. So it's primed and synthesized this way. They flipped it on me, but anyway, it's synthesized in the other direction. So you end up with 2 strands. Right? And you can see here, this guy's been extended by the telomerase, right? And all we've done is added more TTG, or whatever it is, TTG, wait a minute, where's the G? There's a GG. So we've added more of that repeat. You see what I'm adding? I'm adding this repeat that's here, TTA-GGG. Not T, no T. So the TTA-GGG, we're adding that back on. And then once we do extend that, then we can come back with DNA polymerase and read that strand as a template to extend that. So what we do is we maintain those multiple repeats on the end of the chromosomes, that telomere, so they don't stick together. Okay? Question? So what happens if they do stick together? What happens?
Telomerase.
Where do you find? So who knows where telomerase was found? So telomerase was found first in HeLa cells. Who knows what HeLa cells are?
Those are cancer cells.
They're cancer cells, right? They came from a woman named Henrietta Lacks in the 1950s who had cervical cancer, and her doctor took a chunk out, put it in a tissue culture dish, and they're still growing today. And I just heard in the news sometime this week that her family's fighting now because they've sequenced that DNA, so now we know her whole genome. And they think it's rather personal and it shouldn't be out there. So they're suing. But anyway, that's what HeLa cells were. So telomerase was isolated in HeLa cells and had this activity. But when someone started to do this in mice, right, what people did is they took a mouse and they made a knockout mouse that didn't have telomerase in it. And they expected that the chromosomes wouldn't be elongated on the end and the mouse would eventually die, right? They figured something would happen if you didn't lengthen the chromosome. Well, it turned out that the mouse didn't die, and his children didn't die, but his grandchildren died. Right? Why did it take so long? Because remember, those mice had that 60 kilobase pair repeat. This guy here, it's so long, so 60 kilobase pairs on the end. Well, what happens is that as cells replicate, the telomere is shortened, at least they're predicted to shorten by 50 or 100 nucleotides every time a cell replicates in human cells. So if you have 5 kilobase pairs on the end of the DNA in a human cell, if every time it replicates, right, in a normal cell, if you lose 50 to 100 nucleotides, then in about 50 generations, cells die. And in fact, that's what happens. You can scrape cells out of your cheek, put them in a tissue culture dish, and go from dish to dish to dish about 50 times. They'll divide about 50 times, and then they die. They don't go any further. And what happens is they're losing their telomeres. Okay? So once they lose their telomeres, that protein can't bind to them. Their chromosomes stick together. What's the problem when the chromosomes stick together? So if the chromosomes stick together, right, here's one chromosome, and it has this kinetochore where the spindle attaches to, and now it sticks to another one here, right, with a kinetochore of its own, and now these get attached to the kinetochores, get attached to a spindle, so during mitosis they're just pulling, and what happens, these break, and they don't necessarily break here, they can break here, or anywhere. So the ends of your chromosomes fuse, they break apart, and now you have one cell with too much DNA, one cell with not enough DNA, and you end up getting mutant cells and you develop tumors. So what happens is the cells die. They senesce and they die. So normal wild-type cells don't have telomerase. Henrietta Lax's cells, tumor cells do. Tumor cells can grow forever in culture. Hers have been in culture since 1950-something because they keep replenishing their telomeres, right? Where else do we find telomerase? Where else? All the answers. Go ahead. Gametes and early cells, right? Because when you first form a zygote, that guy's got to divide. You got one cell, one fertilized cell, and he's got to go to quadrillions of cells. He's got to divide lots and lots. So during early development, right, cells that are not differentiated in particular have telomerase so that they can replenish and the organism can develop. Yes, ma'am?
So does the telomerase degrade in wild-type cells?
It's not expressed. The genes are just shut off, so they're not expressed. And one of the things that happens in many tumor cells, and I don't know exactly the number, Michael turns upstairs to tell you, but in many tumors, many cells that become transformed, that become grown, will grow and form cancer cells, tumors. In many of those cells, it's like 70% of them, the telomerase is turned back on. Usually tumors have lots of different things, right? They lose their contact inhibition behavior, lots of things. And one of them is that the telomerase is turned back on in those cells, which allows them to grow forever. Certainly Henrietta Lacks is turned back on. So anybody have a question about that? So that's telomerase, that's why we have it. That's why your cells are senescent. You don't have any, why do we want a cell to be senescent? Why does nature design that into a cell? Life of the cell is tough, especially a eukaryotic cell, right? There's these big old ribosomes clomping around. There's proteases and RNases. There's UV light. There's all kinds of damage. So after this guy who's been around for 50 generations or whatever, he's getting old and he's ready to turn over. So that's what happens. Rather than having all that damage build up and be some sort of a mutation the older a cell is, the more likely it is to have a mutation that will have some consequence like weaning that cell from cell cycle inhibition, and it will just divide. Okay, I gotta go on around here forever. We talked about retroviruses. I just want to show you this is an RNA tumor virus. They have a capsid inside that's built with protein. They have an envelope on the outside. The envelope of these is actually the membrane of the cell that they grew in. So this is the membrane of the host cell. The green guys are proteins that are stuck in there, right? They're glycoproteins like GP120 and the AIDS virus. These proteins are the guys that bind the receptors on your cells. And the AIDS virus, this green guy binds to T cells, and that's what gets it into the T cells so it can restart its cycle again. When this binds to a cell, essentially the membrane is lost and you end up with a capsid. The capsid is turned over. You lose the envelope, you lose the capsid, and that lets the genetic material, which in the case of RNA tumor virus is a piece of RNA, the blue guy, come out, and that RNA is now out of the cell. Remember, for DNA synthesis, we have to have a primer. So what this virus does is when he replicates, when he's forming the virus, he takes a host cell tRNA and attaches it as a primer. So the primer's there. This guy opens up, and now there's an enzyme that's called reverse transcriptase. Why is it called reverse transcriptase? It's an RNA-dependent DNA polymerase, right? Normally, when we talk about transcription, we go from DNA to RNA. Now we're going from enzyme called the reverse transcriptase. The reverse transcriptase uses this RNA as a template, tRNA as a primer, makes a strand of DNA. Now we have a hybrid DNA molecule, a duplex between RNA and DNA. There's then a molecule called RNase H, and RNase H will chew back on the RNA, and as it does, the polymerase will, it has polymerase activity, and it will synthesize DNA. This is not a great picture, I don't want to go into it a lot, but it just shows you that this guy has RNase activity and synthetic activity. The foam separates the strands, and what happens is as it chews off this chews off that piece of RNA, this is a template, it adds on the DNA. You don't have to know. All you have to know from this picture is this molecule is a monomer. It's like DNA polymerase 1. It has multiple enzymatic activities. It has a RNase H activity, and it has a polymerase activity. Which one is it missing from, what's missing from it that DNA polymerase has? DNA polymerase 1 has. What's the activities there? Go ahead. It's proofreading. It doesn't have a 3' to 5' hexanuclease, right? So what did I do there? So the question is, if we know all this about these viruses, and we do for other viruses and bacteria, we can develop antibodies. Why can't we develop an antibody to treat a retrovirus? What's an example of a retrovirus? What are some of them? HIV, what else? Hepatitis, what else? There's a bunch of them. It doesn't matter, but there aren't any tumor viruses. Why can't we develop an antibody? Retroviruses of all sorts can continually change their antigenicity. So the proteins on the surface change slightly. They're always the same protein, but they have a little different protein. They change their antigenicity. How? Mutations of their genome. So there's actually mutations of their genome which changes their protein. Is it due to environmental assaults? No. They're mostly mutations in the genome. And where do those mutations occur in that genome?
Because you don't have proofreading. Because there's no proofreading.
They don't have proofreading, right? So during replication, DNA polymerases, actually the DNA polymerase in eukaryotic cells, I don't know if I told you this, but DNA polymerase itself makes a mistake about once every 10 to the 5th nucleotides. Remember I said proofreading improves that by 10 to the 2, about 100-fold. So because of proofreading and how good the eukaryotic enzymes are, they only, in E. coli, they only make a mistake about once every 10 to the 7th nuclei. So that's more, or base pair, so that's more than the genome in E. coli has, right? 3.3 times 10 to the 6th. So that means that you can replicate some of the E. coli genome, some of the bacteria replicate without a mistake, no mutations. But some of them have one or two. But this guy makes a lot of them, okay? So here's the deal. Retrovirus genome